Hello, everybody, and welcome to Building Healthy Soil at Viva Farm. My name is Amy Larson. I'm a researcher here at New Mexico State University at the Sustainable Agriculture Science Center in Alcalde, and I'm joined by Bryce Richard, owner and operator of Viva Farm in Las Cruces, New Mexico. And we're here to talk a little bit about the research projects going on at Viva Farm. So just to give you a bird's eye view, the Sustainable Agriculture Science Center in Alcalde is one of many science centers across New Mexico. We're in the northern part of the state. Bryce Richard and his family are down in Las Cruces, which is also the city where the campus of NMSU is located. So the Richard family own and operate Viva Farm in Las Cruces, New Mexico. It's a multi-generational family farm. They grow vegetables and manage a pecan orchard and raise livestock. So of course they're very busy. The SASC and Viva Farm are collaborating on an on-farm research project funded by a New Mexico zone grant from the Santa Fe Community Foundation. And I'm going to turn it over to Bryce so he can talk a little bit about the on-farm research project.
Thank you, Amy. So we got into this as kind of a continuation from a prior grant that we had gotten for promoting sustainable agriculture in the region, and this opportunity came along as part of the Soil Health Champions Network, and I really felt that we'd be doing a disservice if we didn't jump on this and try and continue that project. So our main goal is trying to cultivate and sell high quality, nutritious food at an affordable price. And we were kind of running into problems with that last part because we were spending way too much on inputs. So that really kind of led us down the regenerative agriculture rabbit hole, I guess you could say. And so now that's what we're doing. It's kind of a definition here that we've put together is that regenerative agriculture is restoring the soil function, so the microbes and everything the way that they should be in order for plants to get the nutrients and water that they need for optimal growth. And as a byproduct of that is improved farm profitability. These are some of our the research project goals. So we really want to try to figure out how to measure soil health and what the impact of our management decisions have on those. So it's measuring soil health can be a little bit tricky because soil is a living dynamic system, and a test is just a snapshot of that particular at that time. So we've got a whole suite of tests here that we're doing, your run-of-the-mill chemistry and salinity, microbial communities of the PLFA, which is phospholipid fatty acids, the soil respiration through the Haney test, labile carbon, which is the new carbon that's going into the soil, and then plant tissue analysis to see what the connections are between what the plants are actually accessing and what is showing up as soluble nutrients in the soil test. So hopefully out of all of these tests, we'll get some good correlations to what we're actually seeing in the field and how the plants are responding. And this is what our management decisions are based on. These are the NRCS soil health principles. So they have 4 of them here. It's minimizing disturbance, and that can be reducing tillage, fertilizers, herbicides, pesticides, maximizing your soil cover, which is a big one here, mainly for conserving water, and because our sun is so intense. That's going to be reducing, usually reducing tillage, but it also comes into play with grazing systems, making sure that they're not overgrazed. Maximizing your biodiversity, whether that's widening crop rotation and cover crops, bringing livestock back into an operation, or multiple species of livestock. And then maximizing continuous living roots. And that's just making sure that there's always something growing out there to feed those microbes and keep new carbon going into that system. And that can be as cover crops, relay cropping. There's a lot of ways that can be addressed. And the main thing to remember is that all of these principles on kind of as individuals and on a whole is going to depend on your context, what your goals are as an individual, grower, your goals long term, and the initial challenges that that you're really running into with even all the way down to specific fields or areas of a field, whether that's addressing poor drainage, salinity, nematode issues. So I mean, you can really tailor each of these to what you're trying to accomplish. And these are kind of a real short version of what we're currently doing on the farm and what we're translating over into this research project. So right now we use diverse cover crops, and the research will be doing a winter cover crop that we're going to be terminating with a roller crimper. And then we do vertical tillage in the spring and the fall, relatively shallow. This is only about 2 inches deep. And that's for planting our cover crop, and then also so that we can plant into it in the spring, since we don't have a no-till planter yet. And then we're using the JSB as Johnson Sioux Bioreactor Compost as an inoculant to increase the soil microbial diversity. And it's important that that this is an inoculant rather than trying to use it as a nutrient source. And that just plays into how this compost is different from what most people would think of. And this is just an aerial view of the farm. It's a pretty gerrymandered property. This was originally all farm ground, probably 100 years ago. And then it's slowly been left for dead and chopped up into housing, and this is basically what remains of it. So the research trial area is in that red circle there. It's just under half an acre. And then to the right of that circle, we're also doing a cover crop trial separate from this project, where we're trying out 28 different either species or mixes of winter cover crops. And then we'll use that to tailor our mixes going forward with this research. And this is a detail of the layout for the research. So we're going to be planting 4 different crops out there for each of those years, for the 5 years. This is just a single replication. There would be 2 more replications of this in that field. And then we're taking yield samples from the middle beds of each of those, so that we can hopefully reduce some of the edge effect between the treatments. So you can see in the treatments there that we have one where it's just a cover crop, one with a cover crop plus the compost inoculant, the inoculant on its own, and then a control, which is just the conservational tillage. So hopefully we can kind of separate out what each of these is bringing to the table, and maybe see some synergism between the cover crops and the compost. And so this is our fertilizer plan for the research. So we use organic fertilizers on our operation. So this one will be using poultry meal. It's a 10% nitrogen. And these rates are a bit lower than what we use on the rest of our operation currently. This is a pretty big cut. And it's based on what some rates that Christine Jones had put out in her paper, Nitrogen, the Double-Edged Sword. And then also on research that Dr. Johnson has had with no results working with other growers and in his test plots. And the goal with reducing them so much is to motivate the plants to form those relationships with the biology rather than relying on the crutch of applied nutrients. We'll really see that in those last 2 years where we're not applying any fertilizer onto that field. And we'll really see what the microbes are able, the microbes in the cover crops are able to supply for that growing season. We start with, we use sprinklers to get our seedlings and transplants started in the first part of the year. And then we switch to a subsurface drip system for the remainder of the season. And this is all controlled by soil moisture sensors and automated valves. In the research plot, it's not marked on there, but we'll have 2 separate irrigation systems on that, with an additional one for the non-cover crops treatments. And hopefully we'll be able to do some comparisons in water usage between those, as I know that's really a major concern with additional plants growing out there, that we might be losing more water than what we're getting back in the season. So it'll be interesting to compare those.
So Bryce had mentioned the Johnson-Su bioreactor compost. So we wanted to take a few minutes to explain what's unique about this compost and what the benefits we hope to see from it are. So the process was developed by Dr. David Johnson, who's a NMSU researcher, and his wife, Wei-Chin Su Johnson. And the basic premise is that this no-turn aerated composting process is designed to really maximize species diversity and numbers of microbes within the compost. And the way they do that is it's first start with a unturned compost system, which is allowed to go through the decomposition and maturation process over a long period of time, typically 12 months. It's best if if the temperatures within the compost pile are maintained above freezing, and that allows the decomposition process to continue throughout the winter. It's also very important to water this compost daily, and that can be done with an irrigation system and a timer. And then once the compost is through the heating cycle, it's a great idea to add worms, and that the worms keep the compost churning. They add a lot of organic matter to the compost and make it even more rich. So very quickly, we start with typical materials that you might find in any compost process. It's important to get them well saturated, to make sure that they're well watered and that there are no dry spots within the compost. And then as the compost is loaded into these bins, there are air vents that are maintained throughout the life cycle of the compost. And here you can see compost that's well along its process in decomposition. And above here, you can see the irrigation system. So there's a sprinkler system that's keeping the compost moist. And so the benefits of this particular process, again, as I had mentioned, is it's designed to allow the microbes time to really gain in not only diversity, but in numbers as well, particularly the fungal diversity, because if you avoid turning the compost, those fungal networks, the hyphae, are allowed to mature, and the fungi can grow, and they're not disrupted through turning. So here's an example of a compost sample, a Johnson-Su bioreactor compost sample that we took to the lab to measure the soil microbial biomass. And as you can see here, they're measuring fungi, they're measuring bacteria, they're measuring protozoa, including flagellates and amoebae. And all of these numbers are either within the desired range or above, which is very promising and hopeful that these microbes that are allowed to grow and expand in diversity in the compost, then once they're applied to an agricultural system or a garden, for example, that they will form those plant microbe associations and really inoculate, continue to grow within the field soils and help the plants in accessing nutrients and water, what Bryce had mentioned earlier. So moving on, we wanted to kind of get a sense, have a short discussion with Bryce about what it means to have an on-farm research project while trying to run a business. And so the first question I had was, what have you learned from the project this far, and what challenges have you encountered in trying to do this research project?
Well, what I've probably learned the most about so far, I mean, we're very early in this project, but mainly the different tests that are out there, and then how to use and interpret those different tests, what each of those is telling you about that soil. So some of those I had heard about, but I'd never used before, such as the POXC, the PLFA. We've used the Haney before, but I didn't fully understand how to interpret it. So I'm really excited to be using a lot more of those tests, and then having the ability through the funding of the grant to be able to run all of those on a single sample and see how they compare with what we're seeing in the field. And probably the biggest challenge is actually collecting all the samples for those tests. Because when you're doing a replicated trial with 4 treatments on them, and you're doing 12 different samples for all of that, and it can be a little bit tedious to keep it all labeled and sorted out. So, but I guess the main thing to remember is that you can do on-farm research, and it doesn't necessarily have to be replicated. It's not something you're going to be publishing, but it's important for you to be able to keep it manageable for what you can handle, and still be useful information for what you're trying to gain out of it.
Great, thank you. And second question, can you offer any advice to farmers who are interested in improving their soil health, but also worried maybe about the risks and concerned about maintaining profitability on their farm?
Well, I think the first thing would be to start small. Anytime you're doing something new, you really don't want to risk the whole operation on it. This was some numbers that were put out by Bob Recker. He's does research now, but he used to work for John Deere in their R&D department. But what they would do is you had 10% of your acreage would go into general research, something you think is going to likely going to succeed. Maybe it's a small tweak to your equipment, a new cultivar, different species, something like that. But you're pretty sure it's going to work, and then you can roll that into the rest of your operation if it's successful there. But out of that 10%, you use 1% to really try out something that you have no idea if it's going to work, if your equipment's going to be able to handle it. And that might be if you're doing something like no-till, your cover crops, where you're really not sure how it's going to play into your system. And if it's successful, then you can scale that up into that 10%. And if it's a flop, then you can analyze, why did it not work? Was it timing? Is there something that I can tweak that will make that successful?
Well, thank you so much. And I think that, innovation and learning always requires people out there on the forefront really trying these techniques out and trying new strategies. And so what we hope to do is share our experiences, your experiences through this process with everybody in New Mexico. And so hopefully we can put on more of these videos and share what we learn as we're going, and hopefully folks can get new ideas, try things that they hadn't tried before, and we can build a support network of people doing this regenerative ag projects. So thank you so much. And just to close out, I wanted to bring up some of these soil health resources, websites, organizations. There's certainly a lot here in the state of New Mexico that I wanted to highlight. It's certainly not an exhaustive list, but hopefully through these websites and other research, farmers can get the information that they might need in order to get started. So if anyone has any questions, Bryce and I are certainly willing to answer anything that we can answer, or find out more information if we don't know the answers. And we're more than happy to support all you guys out there in New Mexico trying to do right by the land. So thank you so much, and thank you for attending this presentation.